Hi, my name is Aniket Ramshekar, and I'm an MD-PhD graduate student in Dr. Hartnett's lab, and today I will be presenting our poster titled Identifying Sex as a Biologic Variable in Tamoxifen-Inducible Cre-LoxP Knockout Mice. Neovascular age-related macular degeneration, or neovascular AMD, is a leading cause of vision loss worldwide. The burden of vision loss is reported to be higher in women than men in age and gender-adjusted epidemiologic analyses of data from the World Health Organization. Several factors may be involved, including not only access to care, but also potential biologic differences based on sex and gender. Here, we study the pathology of neovascular AMD, and in particular, the signaling mechanisms involved in a key step associated with vision loss, namely the activation of choroidal endothelial cells to migrate across the retinal pigment epithelium into the neural retina to form choroidal neovascularization. We found two proteins, IQ motif-containing GTPase binding protein 1, also referred to as IQ-GAP1, or erythropoietin receptor, also referred to as EPOR, to be important in angiogenic processes such as migration and tube formation using cultured choroidal endothelial cells from human donor eyes of both sexes. We addressed the hypotheses that EPOR or IQ-GAP1 would increase neovascular AMD in males and females by using experimental models of laser-induced CNV with choroidal cell-specific knockout of either IQ-GAP1 or erythropoietin receptor. We evaluated results generally and based on the sex of mice. To address our hypotheses, we used both male and female endothelial IQ-GAP1 knockout mice, endothelial EPOR knockout mice, macrophage-specific EPOR knockout mice with respective littermate controls that have no alleles of either Cre. All mice were on a C57 Black 6J background, and genotypes were validated by transgenics using qPCR. Once mice were 4 weeks of age, we administered intraperitoneal tamoxifen every other day for 3 injections. 2 weeks following the last dose of tamoxifen, we performed a laser-directed injury to induce the development of CNV in mice. 1 week after laser, we collected eyes and stained with lectin to visualize CNV in the posterior eye. Lectin stained lesion volume was measured using a computer software and verified by hand by a master reviewer. We performed a multi-level linear regression model to statistically analyze the results. What we observed was when we knocked out IQ-GAP1 and endothelial cells, there was a significant reduction in laser-induced CNV, but when we stratified the data by sex, significance was only observed in male and not female mice. When viewing the data, the lack of significance in females appeared to be related to the effect of tamoxifen in reducing CNV in non-Cre control female compared to male mice, and that data is summarized in Figure 1. When we knocked out erythropoietin receptor in endothelial cells, there was no significant reduction in laser-induced CNV, but when we stratified the data by sex, there was a significant reduction in laser-induced CNV in males, but not in females. There was also a non-significant trend of reduced CNV in tamoxifen-treated non-Cre control female compared to male mice, and this data is summarized in Figure 2. When we knocked out EPOR in macrophages, there was no significant reduction in laser-induced CNV, even when we stratified the data by sex, and this is summarized in Figure 3. There was also no effect of tamoxifen on non-Cre control female compared to control male mice. To summarize, our data suggests that endothelial IQ-GAP1 or EPOR knockout reduces choroidal neovascular or CNV volume in tamoxifen-injected male mice, and that macrophage-specific EPOR knockout does not have an effect on laser-induced CNV volume in either male or female mice. Analyses of experimental models by sex is important to determine potential biologic differences that can be difficult to sort out in population-based studies of humans that account for gender. This study highlights several points. It is important to use non-Cre tamoxifen-injected controls when studying cell-specific knockout mice in order to assess possible effects of tamoxifen on sex of animal. The differences in effects seen in the 3 different models may be related to the mechanism of pathology, or the mechanism only has an effect in only one sex. The data we present today, however, has not been specifically justified for sample size based on sex, and the data may change once sufficient sample sizes are tested. We thank you for your interest in our study and look forward to the opportunity to answer any questions. Thank you.